shaped bacterium. And then all these little dots are bacteriophage, which are viruses that lyse bacteria. So there are some animal viruses like polio that works that way. But most animal viruses actually get out through budding. And I think that's actually like an advantage, because if they get out through budding, the immune system might not notice as much. Because if you explode a bunch of cells on the way out, you're going to get the immune system's attention, and then the nosy immune cells will come over and see what's going on. So I think it's a way to be latent, and it's a way to avoid the immune system if you get out of the cell through budding, because then you're like kind of under the radar. All right, so bacteriophage are viruses that infect bacteria, and basically there's different parts to their life cycle. The reason we kind of mention them is because they are responsible for mutations that occur in bacteria. So that was called transduction, when you get genetic material from a virus, and that's what can happen to some of these bacteria, is that they'll land on the bacteria and they'll basically inject them kind of like a syringe with genetic material, and then that takes over the host chromosome, and then they use the bacteria as a replicating like factory, and they make a bunch of copies of that virus. So there's two parts to the life cycle of the bacteriophage, which is called lysogenic and lytic. When they are integrating the genetic material into the host, but the cell lives, then that's the lysogenic cycle. And so that's where new genetic information can be picked up by the bacteria, because they have mutated the chromosome. So some of the viral DNA has ended up in the chromosome DNA that belongs to the bacterium, and that can be stuff like antibiotic resistance or heat resistance or toxin production. And so then every cell after that will have that genetic material. But sometimes the virus will infect the bacteria and it'll actually want to replicate itself. So it's not in a quiet phase, and that's called the lytic cycle. So the lytic cycle is going to result in death of the bacterial cell, because as part of that process, the virus is going to actively start replicating, and when it tries to release, then it'll kill the bacterial cell. So those are the two parts of the life cycle for bacteriophage, which are viruses that infect bacteria. All right, you have to be able to give examples of all different kinds of viruses. So I said you had to know like examples of an RNA virus and a DNA virus, and then an example of an acute, persistent, or latent virus. So acute means rapid onset and a quick resolution. So more like on the scale of like days. And so that would be stuff like cold and flu. And sometimes it might even be subclinical, like you didn't even know that you had anything. Now Ebola is also considered acute, but you might die from it. So acute doesn't mean safer necessarily, although in general it probably does. Like most things can't kill you like super quickly. So acute viruses are ones that act quickly. Persistent is the same as chronic. So these are ones that can persist for months and years potentially. So classic examples like HIV, apparently measles and rubella can, because they can be latent, I guess, and that's why vaccination is important. And then hepatitis is a good example of a latent virus, because those viruses can hang out in the body indefinitely. And so it can be fatal then, because you have damage like accumulating over time, but it depends on the virus. And then latent is one that's really interesting to a lot of students, I think. That's the viruses that reactivate. So those are things that you carry in your body, and if you get stressed out or your immune system goes down, then you're able to have that virus sort of reactivate and come out again. So classic example of that is shingles and cold sores. They hide in the nerve, the viruses hide in the nerves, and then when conditions are right, they will emerge again, and so you'll see them go up again. So there is one virus that is an example of all of these, and that's HIV. So HIV does have an acute retroviral syndrome where when you first get infected, I think it's 70% of people will have a flu-like illness. So they'll get, within the first couple weeks to couple months, they will get sick. But then their immune system will kick in. They'll start making antibodies. And if the T cell count stays high enough, it'll just go to a chronic phase where they have essentially latent HIV. And then as the immune system breaks down, the viral load will go back up, and then it'll reappear with the signs and the symptoms. So HIV happens to be a good example of all. Is there anyone else that needs the notes? Okay. I saw you didn't have them. I didn't want to call you out awkwardly, but there you go. You're welcome, Kylie. Okay. Sorry if I keep pulling down like this. My stupid glasses are fogging up, so I'm trying to vent. So hopefully that's not distracting you too much. Normally I wear my contacts, but my eyebrows are all crazy right now too, so I'm just feeling self-conscious. So I'm don't look at me. Don't look at me. Okay. All right. So there are viruses that cause cancer, and those are called oncoviruses. And this is also another recent discovery that's pretty interesting. So I think I actually was reading now that they know of 50 viruses that are linked to cancer, or 50 cancers that are linked to viruses. And it's the way that the viruses take over the whole cell machinery that causes it. So they've actually identified, on HPV as an example, and that's what they're showing on the right side over here, is that the genome of HPV is like a circular chromosome, and there are these certain proteins on that chromosome that allow it to integrate into the host chromosome. So it actually opens up that ring, and then it allows it to become part of the host chromosome. And so that is going to allow it to persist because the information is there, so it can reactivate pretty much at any time. But also it's going to cause mutations in that DNA sequence because that's not the normal sequence, right? And if those mutations happen to be in cells that in genes that control the cell cycle, then that's when cancerous changes are possible. It doesn't mean that you will get cancer if this happened, but it just increases your risk of getting cancer. So how do they cause cancer? Their role in cancer is considered multifactorial. There's more than one thing that has to happen besides the virus, that would cause cancer. So typically the viruses will be considered either a direct or an indirect carcinogen. Carcinogens are things that contribute to cancer, and the ones that are direct is where you can find bits of the virus in the tumor, for the long term, which links it to that cancer. But then weirdly, there's some viruses that disappear after a while. So once the cancer starts growing on its own, the virus isn't really, it's autonomous. The virus isn't really triggering that anymore. In addition to the virus itself mutating the DNA, there are other things that happen that lead to a cancerous phenotype, which is being shown here. So viruses can evade the immune system. And so by evading the immune system, it gives them the ability to become latent, meaning that they can hide out for a long time, and then those changes in the DNA can start accumulating. And all cancers, the hallmark of a cancerous cell is that it has an unstable genetic makeup. And so the longer those viruses are in you, the more changes can pile up. And then since they can avoid your immune system doesn't really know what's happening, and so they might not deal with it until some changes have already occurred. The other thing that causing these mutations, the virus, when they cause them, it causes immortalization of the cells. So normally cells only have a certain lifespan. They only divide a certain number of times before they are kindly asked to leave. It's programmed cell death, which is called apoptosis. But viruses can mutate the genes that are responsible for apoptosis. And so cancer cells evade apoptosis. Especially the ones that have been infected by a virus, they can avoid the death penalty. on them. So even though they're abnormal, they'll keep growing. Chronic inflammation, viruses can cause it, and chronic inflammation is linked to cancerous changes. Because of the chemicals that get released as part of inflammation, they are, I guess, interfering with the cell cycle in a way that can lead to tumor production. I mentioned the initiation of cell death. That can be avoided by a cancer cell, but the cell death that the viruses cause can also exacerbate the inflammation, which then exacerbates the chance of cancer growing. So an example of this, like in HIV, it actually induces cell death in cells. And then that causes more immune cells to come over, and then those cells get infected. And then those get destroyed, and then more come over, and those get infected. And that's how HIV is able to maintain itself in the host. So damage to the cells, cell death due to a virus, will trigger inflammation that can contribute to cancer. You block the tumor suppressor genes. So in the cell cycle of the host, there is checkpoints that say we cannot go past this point until a certain condition is met. And it's usually DNA related, like the DNA has to be proper. So the tumor suppressing genes are like stop signs. So if you remove the stop sign, you suppress the suppressor, then the cycle is always on. And then that leads to uncontrolled cell growth, which is basically what cancer is. And then the environmental mutagens. So it's not just the virus and the mutations. There's usually other environmental factors like smoking, hereditary things, diet, exposure to chemicals at work. There are other things that can contribute to cancerous changes. So cancerous changes are due to many mutations over a relatively long period of time. And things like HPV and other cancer-causing viruses can cause those mutations to add up more quickly. All right, so this example here is actually really interesting. What they did was they found 796 archival cervical biopsies. So these are ones that have been preserved. And they wanted to see which genes are overexpressed if you're moving towards a cancerous phenotype of those cells. Because cancer has a genetic component to it. Like everyone's tumor has certain genetic markers to it. And so one of the things that I guess was hypothesized was that one of the oncogenes for which is amplified when the cell cycle is out of control is this protein that's called P16. So they wanted to see, is P16 elevated in people that are progressing towards cancer? So they took these samples of cervical cells that were at different stages of dysplasia. One of the ways they grade dysplasia is called cervical intraepithelial neoplasia. So the cells are changing, and it's level 1, 2, 3, and then cancer. So what they did was they used antibodies that were labeled with like dye that would bind to P16 protein if it was in the tissue. And so then you would see this dark red color. So then what they saw was that in normal tissue, there is no like detectable P16 because P16 should normally be low if the cell cycle is moving the way it's supposed to. Then as you get to a higher grade lesion, more and more of the P16 is showing up. And so it's just again reinforcing for them this idea that in cancer cells, P16 is overexpressed. And so this would be what you call like a biomarker, which are certain tests you can do that give you evidence of disease potentially. It could be like a screening test of some sort. Then they wanted to link the production of P16 to the strains of HPV. They were wondering, like, okay, do all HPV strains cause an increase in P16? And I think I mentioned before that there are high-risk strains of HPV. Those are the ones that cause cancer. And then the regular strains are the ones that can cause genital warts. They're not the same. So just because you have genital warts doesn't mean you're going to get cervical cancer, and vice versa. So they were lucky enough to find a sample in their little record that actually had 2 different strains of HPV in it. So this was a person who may or may not have had genital warts, but HPV-6 is a strain that's associated with genital warts. And then HPV-16 is associated with cancer. So the first thing, so this procedure is something called in situ hybridization. Basically, what they can do is look for HPV genetic material using a probe that has nucleic acids that have the opposite pairing. Like, so if they're looking for a little probe that has a certain sequence, they can find it by having the opposite sequence like bind to it. And then it also has dye indicators, so that tissue will turn a color if that genetic material is in the tissue. And so that's how they were able to figure out a piece of tissue that had 16 and had 6 in it. So then they took that same tissue and they did the in situ hybridization to look for P16. And what they saw was that the tissue that had HPV-16 in it was positive for P16, and the lower risk strain, which is 6, was negative for P16. So that's again linking that there are strain differences. There's like 100 strains of HPV, but they don't all cause cancerous changes. But this is reinforcing that HPV-16 can cause cancerous changes. And they can see that by the surrogate of P16. If P16 is elevated, it would mean you're more likely to have a high-risk strain of HPV. So that's kind of a lot, but does that make sense? That was like pretty cool. I was like, oh, all right. So that's really just, I think, supporting that HPV causes cancer. Like, we're pretty sure. In addition to the other things that contribute to cancer, like chemicals and stuff like that. All right, so again, how does the cancer happen? Like, what's the deal? So normally, in a eukaryotic cell, cell division is very tightly regulated. So the cell cycle spends most of its time in sort of a quiet phase. So there's like G0, which is just like where it's just hanging out. And then there are gap phases, and those are the checkpoints, where before the cell cycle will proceed, it has to get past that checkpoint, and those checkpoints revolve around checking the DNA, like proofreading it. So very little of the cell cycle is actually mitosis, which is where you have the prophase, metaphase, anaphase, telophase, and the chromosomes are going to separate, and you're going to end up with 2 new cells. That won't happen unless the DNA is like all perfect. So the genes that typically regulate this are either called tumor suppressors or oncogenes. The tumor suppressor is the stop sign. It's the red light. So if you get a virus and it mutates a tumor suppressor gene, then the stop sign is removed. The red light is removed. Imagine that you took an on switch and you just put it in the down position, like just removed it, like so that it cannot be turned off. That's what happens when you mutate a tumor suppressor gene. You suppress the suppressor, you remove the stop sign, therefore it goes. So the cell cycle will go even though, and that's because the stop signals are missing. Now on the other side, I always remember oncogene on. If the oncogene is overexpressed, then the cell cycle stays always on. So imagine a light that always is green. If the light's always green, the cell cycle is always going to go. If the on switch is always on, so imagine you super glued the light switch into the on position, then it's always going to stay on. And so that also has the effect of potentially cancerous growth. Because in both instances, you are mutating the genes that control the cell cycle. You're either removing the stop sign, and that's loss of a tumor suppressor, or you're overexpressing the go sign, and that's where oncogenes get mutated. And so this is due to the fact that viruses, when they replicate, they incorporate themselves into your genetic material. Like they literally become part of you, which is creepy. And then that can cause mutations that lead to this dysregulation of the cell cycle. So here's an example of something called Merkel cell carcinoma, which is a type of cancer. This is a relatively recent discovery in 2008, where they figured out that this particular skin cancer was linked to this virus that was called Merkel cell polyomavirus. So apparently this is just one of those weird viruses that's like floating around, and you get exposed to it as a child, and I don't even think it causes like clinical infection. Like I think it just is like there. And there's a lot of viruses like that are just kind of quiet. They don't really do anything. But the virus goes latent. And then as you get older, or if you got a disease or your cancer treatment or something else going on, you can become immunosuppressed. And it's that immunosuppression that is kind of like the first chance for the virus to start replicating again. And then it opens itself up and integrates it into the host chromosome. So that's the first mutation. And then if you couple that with the fact that this is happening in the skin, and you get exposed to UV radiation in an excessive way, that creates other mutations so that you're more likely to have this particular cancer that's called Merkel cell carcinoma. And I don't know much about Merkel cell carcinoma. Like, I'm not sure if it's like aggressive. I mean, it looks like it traverses pretty deep, like it goes across the dermis. So that's due to this virus. So the other thing you have to know examples of, and again, it's kind of your choice, of viruses that cause cancer. So if you say that, like, if you don't know that HPV causes cervical cancer after this, then I would be sad. Sometimes people still get that mixed up. Or maybe they don't know what the cervix is. It's the bottom of the uterus. So HPV is linked to cervical cancer, anogenital cancers, penile cancers, head, neck, and throat cancers. So they've linked it to a lot of different cancers. And if you really think about it, it makes sense because of how it's transmitted. Okay, it's transmitted through sex. And so it makes sense that those are the areas that it could colonize potentially. Epstein-Barr virus, which is what causes mono, that's linked to this lymphoma that's called Burkitt's lymphoma. Hepatitis B and hepatitis C are both linked to liver cancer because the inflammation from them can cause cirrhosis, and having cirrhosis increases your risk of developing liver cancer. This HTLV virus, human T lymphotropic virus, causes adult T-cell leukemia. I think it's another one of those viruses that you just get infected with in childhood and sometimes can reactivate, and then if you have certain genetic predispositions, it can pop up as a cancer. I mentioned the high-risk HPV strains, so not all the strains, but just like the high-risk ones, like 16 and 18. Kaposi sarcoma is a human herpes virus number 8, and that causes Kaposi sarcoma and a lymphoma that it basically attacks the lining of the blood vessels, and that's why you would see people that have AIDS sometimes have those purple lesions, and that's due to a cancer because their immune system is not able to deal with the virus that causes that. And then Merkel cell polyomavirus is a skin cancer that's due to this particular virus. There are other ones. This is just a sampling of viruses that are linked to cancer. Okay, any questions about viruses? Coronavirus. Didn't like reading that. I was telling, yeah, Cindy. What was that? Oh, lupus is autoimmune. Yeah. So you're, let's see, I'm trying to remember. You make antibodies to your own tissues. So one of the tests for lupus is to look for ANA, which are autonuclear antibodies. And so that causes you to make antibodies to a wide variety of your own body tissues. And it does manifest as an inflammation, right, which makes it seem like it's an infection of some sort. But it's actually your own immune system, which sucks. And I don't think they know exactly what causes it. Like, it's not genetic per se, but women have it more frequently. And lupus usually has the biggest impact on the kidneys and like the skin and joints and stuff. So, all right, chapter 14 is all about disease transmission and public health and epidemiology. So It's about how diseases are transmitted and then how we can prevent that. And then it's also about classifying disease. So how do you describe what kind of disease you are dealing with? So that's the gist of chapter 14 is the public health, epidemiology, and then classifying disease. Like how do you put it into a category? So there's a ton of objectives, which we will now begin to go through. Okay? All right, so there's a lot of definitions in this chapter. So if you're someone who likes flashcards, I guess, like this would be maybe your time to shine. All these like terms, right? Like just straight up definitions or things that you just have to know to be able to talk about these things appropriately. So one of the things I find really interesting about micro is that like there's like variable relationships that we can have with microbes. Like most of the time, they're actually like just, it's neutral relationship. And that's called commensalism. Commensalism is like the meh. Like, so you see the little face in the middle? It's like the other one might be benefiting, but the other is unaffected by that relationship. When you're both benefiting, that's in green, and that's mutualism. So mutualism is a mutually beneficial relationship. You're both getting something from it. And then parasitism is what we talk about in here a lot because we're interested in things that cause clinical disease. We're interested in pathogens, and pathogens are parasites. So the relationship that we have with pathogens is called parasitism. And a relationship between two things, a long-term association, is just called symbiosis. Okay, so it's a symbiotic relationship. To say a symbiotic relationship is a little bit redundant because symbiotic means relationship. Okay? So how are you together? You either both are happy, one of you is happy and the other one isn't, or both of you, one of you is happy and the other one is, or whatever. Like, you see what I'm saying? Like different relationships. Okay, so the normal microbiota, the normal flora that live on our body are primarily commensal organisms. So we're not sure what they're doing. But they're not harming us. They're just like hanging out with us. But then some of them are good. Like they're actually doing something beneficial for the host. And we've now discovered that there are good microbes that are critical for health, especially for digestive health and the way the immune system functions and things like that. So which microbes live on your body depends a lot on food. There are certain parts of your body that have more food that are nicer to live. So if you think about your body as like an ecosystem, what part of your body would you consider like the desert? You think about what a desert's like. What part of your body seems dry? Your skin, right? Like your skin and your hair and your nails. And then what parts of your body might be more jungle? Besides like the love jungle. Like let's not go there, but digestive system, right? Like there's tons of food in there, and so there's like tons of competition. And so that competition is called microbial antagonism. The ones that can outcompete will exclude the ones that can't compete. And that's actually a way to protect us because a lot of pathogens that we come into contact with, they are antagonized by the bacteria that normally live in us. And that's how they protect us, is that they are better suited for our body than the pathogen is. And so they're able to keep them out, basically. Okay. So they discovered that these good microbes, the normal microbiota, have very critical health functions. They help with digestion. They help train the immune system. And it's a really fine balance because what can be normal for one person in a certain situation can become pathogenic in another. So example, like some people just carry MRSA. Like it's just in their nose. And if it's just hanging out in your nose, it doesn't hurt you. But if it gets into a wound, then it could. So there's a lot of like dynamics with their relationship that we have with these microbes that live within us. This chart here is summarizing some of the common bacteria that are found in your body just naturally. And so you don't like memorize this chart, but you would just like be able to give examples. If you were asked, give an example of a bacterium that's commonly found as normal flora in the body, you could choose from these, okay? So anywhere it says plus, that means there's a lot of that bacteria there. And then anywhere it says plus or minus, it may or may not be there. So everyone's different. Some people's body would have it and some people don't. And then a star is by the ones where it is potentially a pathogen. And that is an important thing to remember, like I was just saying, that what could be a pathogen in one person might not be in another. And so there are people who exist in a carrier state, where they're not harmed by that bacterium, but the problem is that they are a source of infection in the population. And so they could pass it to someone that's susceptible. So starting from staph down to strep pyogenes, those are all gram-positive. So notice that your body is covered inside and out with staph epi. But notice that staph epi is not a pathogen. It doesn't have a star by it. It's supposed to be there. Like you're literally covered inside and out with staph epi. But staph aureus has a star by it because it is potentially pathogenic. But it's weird because you still have a lot of it on your body. So again, there's just like these interesting dynamics at play. It is potentially a pathogen, but it won't be unless it's in the right situation. And that's what we call opportunistic pathogens. So it's normal for you to have strep in your body. But the problem is that strep is potentially pathogenic. So let's say you have strep pneumoniae in your nasopharynx, in your nose and throat. If you get immunocompromised or you get some kind of weird injury, what if it leaves your nose and throat and goes to your lungs? Then you have pneumonia, right? So a lot of these are harmless in their current setting, but if they move somewhere else, it can cause issues. So from staph down to strep pyogenes, which is what causes strep throat, those are gram-positives. And then Neisseria is always a pathogen. It's not supposed to be there. But Neisseria, E. coli, Proteus, Pseudomonas, and Haemophilus are all gram-negatives. So those are gram-negatives, and they have a slightly different distribution if you look across. E. coli, of course, is going to be, most prevalent in the gut, like in the digestive system. You're going to have tons of it there. And not all E. coli is created equal, because remember we've been talking about strain differences. Some of them are pathogenic, but some of them are just regular old E. coli that's found in your colon. The last two down here are gram-positives again. So Lactobacillus and Clostridium are gram-positive. And Clostridium is potentially pathogenic. So the classic example of that is when we talk about C. diff. You have sometimes small amounts of C. diff in your mouth and your colon. But as long as it stays small, it doesn't really do anything to you because the other bacteria keep it from overgrowing. But let's say you're on a broad-spectrum antibiotic for some reason for a long time. You will disrupt the normal flora, and then C. diff can overgrow in the colon. And then that's when you get things like the pseudomembranous colitis and the extreme diarrhea because you've unbalanced the normal flora. If we had the ability to test everyone in here for these organisms and the different areas, a different pattern would emerge for everyone. And it's very unique to you, but there would be things that are probably the same. Like I guarantee we all have E. coli. I guarantee we all have staph epi because those are things that are like ubiquitous. They're like everywhere. Okay. It's also worth noting that there are some parts of the body that are sterile. And those are areas that are not supposed to have any microbes. So that would be your blood, your lymph, and your internal organs. Okay, like vital organs like heart and brain, kidneys. You are going to have bacteria in your stomach. But if you think about it, your stomach is that link to the outside because your digestive system is basically a big tube from your mouth to your anus. Okay. So they've been studying this for the past 13 years now. I know when I first started grad school, this was like a hot topic. Was this human microbiome project. And so what they did is sample these body cavities to try to characterize which microbes are there. And they can't all be cultured So they had to do molecular methods. They had to analyze the genomes and compare it to a database to see what was there and what things were new. And really, what you would want to do with this information, I think, ideally, is use it for some kind of like medicinal purposes. So like they've identified interesting things about certain gut bacteria being more prevalent in people that have depression or more prevalent in people that have diabetes. So then is there a probiotic we could give you someday? Like if we know exactly what mixture of bacteria would make you feel better, it could be possible. I think we've talked a little bit about C. diff before and the fecal transplants that they can do. They've seen that sometimes you can replenish the good bacteria by having a fecal donor that has a healthy set of bacteria. But then the unexpected side effect they noticed was that when you transferred it from a person that was overweight to a person that wasn't, that person like gained weight inexplicably, like gained a little bit of weight. And so they think that these gut microbes especially control a lot of different things like metabolism and mental health and all kinds of things. All right, so some of these normal microbes can be opportunists. Because like I said, if they get into the right situation, then they might be able to cause harm. And these are just, an opportunity is like some kind of breakdown. A lot of the times it's a breakdown in a physical barrier, like you have a cut or a surgery. But then it's also often a breakdown in the immune system. So immune system and physical barriers are important for preventing opportunistic infections from having an opportunity. And then there's other things that can cause it, like if you accidentally transfer microbes from one part of your body to another, that's called auto-inoculation. Kids do that a lot because they're gross. So they take their little funky hands and put it different places. So like if they have something like impetigo, which is a contagious skin infection, they can move it around. They can move ringworm around, I think, too. So they carry stuff on their little hands and under their nails. Introducing it to a sterile part of your body. So that happens sometimes like if people get, say, a UTI. The upper urethra is supposed to be sterile. The lower urethra is going to have bacteria around it because you have skin there. And so that's why, like especially for women, they teach you to wipe front to back because you're trying to prevent you from accidentally pushing bacteria up into the urethra. But getting a UTI is not necessarily a hygiene issue. Like it's just the fact that you literally have bacteria outside of your urethra. So there's a chance for them to make their way into a place they don't belong. And that's what can cause like a UTI. Healthy carriers. So these are people who just have that thing in their body and it doesn't do anything to them, but they can pass it to grandma or they can pass it to the baby. And then the balance of normal flora being disrupted. That is now recognized as a potential cause of opportunistic infection. Like I said, the case of C. diff. You break down the good bacteria and C. diff can flourish. Another example is like sometimes when women are on strong antibiotics, they can have a yeast infection as a side effect because it kills the good bacteria and then lets the yeast overgrow. Okay, so then there is a difference, of course, between a professional and an opportunistic pathogen. I would say that the majority of pathogens are opportunists because you're going to take the path of least resistance, right? Like, you're not going to try too hard, right? You're going to try hard enough, I guess, to do your thing. But then there's those type A personalities that always do everything perfectly and they try everything really hard. And I think of those as the professional pathogens. So, like, I don't know if you ever had anyone in your class who was, like, always dressed really nice. And you're like, dude, like, come on. And, like, I'm in my pajamas, like, judging myself. So they're the professionals. They got their briefcase. Or maybe did you have anyone who took a briefcase to school? Did they take it that far? So that's fine if you want to do that. I'm not judging. So professional means that you can cause disease no matter what. And if you were to think of a professional, you should think viruses. Like most viruses are professional because they're obligate intracellular. They have to get inside. And so most of the time, if they can cause disease, they will. Most things, though, like a lot of bacteria, are kind of in the middle. So those are called facultative pathogens, where they could cause disease. They usually don't. It kind of depends on the circumstances, but they're more likely to do it than an opportunist. So an opportunist is really a thing that is not that virulent, but if it gets the chance to do something, it can. And some things are like professional opportunists. Like I would consider Staph aureus to be a professional opportunist because staph infections are super common. Like for it to be sort of innocuous, it sure does cause a lot of problems potentially. All right, so these are some common opportunistic pathogens, and you'll see that I color-coded them. Staph is gram-positive. Pseudomonas and E. coli are gram-negative. And Candida is yeast. So gram-negative or positive doesn't technically apply to it. So staph is just all on you. Like it's all over your skin and your body. So it's pretty easy for it to cause an infection. It can get down into a hair follicle. It can get into an incision. It can be passed from inanimate objects, so it's not impossible to get Staph aureus at all. Candida, you don't really get from other people. It's in your own body. And so if it's given a chance to overgrow, then you can get things like thrush and yeast and vaginal yeast infection, or skin infections too. Pseudomonas is one thing that is also on your body, but it's also in water. So, like the craziest Pseudomonas story I ever heard, and this actually made sense because, like, okay, so I'll back up. So, one day I was at the mall with my friend when malls were still a thing, and she's like, "Dude, let's get a piercing, like at Claire's." Terrible idea. And I was like, "Sure." So I got like a like this pierce, like the cartilage at the top, and I was like, "Oh, it didn't even hurt, and it looked really cute." Well, it got infected, right? And I was taking care of it, but I think what happened is that the cartilage broke. And then it just never healed. Like, so when you use like the gun, it like crushes it instead of like poking a hole through it. So if you're ever gonna get a piercing, just go to a piercer, like a tattoo place. So the doctor was like, "You need to take it out." And I'm like, "No, it looks so cute." I got this like big cauliflower ear. He's like, "No, take it out." I'm like, "All right." So I take a strong antibiotic, like because I think it was probably a gram-negative infection actually, like because I have a feeling it was from like just normal like shampooing, like and it getting wet and then underneath there. So luckily I took it out so I didn't like lose my ear. But then I started researching about, they call it chondritis, which is where the ear cartilage gets all inflamed. And I started looking it up and I came across this crazy story where this girl had gone to a salon and she had a new piercing in her ear, like on the cartilage, same thing. And you know how they use the water bottle to like spray your hair, like when they're gonna do stuff? So she ended up getting this like crazy massive ear infection after she went to the salon. And when they like talked to her, or whoever it was, like they did some kind of contact tracing, they noticed, they went back and tested the water and it had Pseudomonas in it, which isn't weird at all. Like Pseudomonas is just in water. But if you have an open wound, like a tattoo or a new piercing, then you don't want that stink water in there. Like, so you want to clean with like the solution they give you, like the salt water solution, or things that will not promote the growth of microbes. So that was just an interesting thing. I didn't even know that was like a problem. And actually then, coincidentally, I had also gotten my hair dyed after that, but I don't remember if they sprayed the water or not. But anyway, I took the piercing out and I haven't redone it yet. So that was just interesting. I didn't even know Pseudomonas could do that. I was like, you can do that? Also, like funky hot tubs, like make sure hot tubs like have proper chlorination because Pseudomonas can be in those. Have you ever been in a hot tub without the bubbles? I'm like, that's just soup. That's gross. I'm like, turn the bubbles on. Anyway, I must sound delirious, right? I'm like tired. It's the end of the week. I'm like, Rita, stand-up time. Okay. So Pseudomonas is funky. It's around. It can cause wound infections. And then E. coli is also just around. They actually do test drinking water for coliforms and E. coli because agricultural runoff, or I guess your well, if it got contaminated, which is not very likely. But if there's like a storm event too, sometimes you'll have an influx of contaminated water, and they look for things like that because it can cause gastroenteritis, but other infections too. All right, so we're talking about first the relationships between us and the microbes that are around us. And now we're talking a little bit about how, when we do have a disease, how do we classify it? How do we put it into a group? So typically there's two big categories that diseases would fall into, and that's communicable and non-communicable. If you look at the word, doesn't it sound kind of like communicate? So communicable are diseases that you can communicate to each other. So those are diseases that require that you somehow are exposed to someone else that has that disease. But it doesn't have to be direct contact. Because as we know, some diseases are vector-borne, right? So like, you don't get malaria from someone else, but if the mosquito bites you and then bites someone else, it can get passed that way. So communicable diseases can be directly or indirectly transmitted. And if they're called contagious, then that means they're very easy to get. They're more transmissible. So what are examples of communicable or infectious diseases? This is like a slow pitch, like easy question.
Like the flu?
Yeah, literally anything you can think of, right? Like the flu, cold, chlamydia, gonorrhea, herpes. What was that?
Stomach bugs?
Yeah, stomach bugs. So like, what is the stomach bug? The
Rhinovirus?
The rotavirus and norovirus. Yeah,
Yeah.
I was having a brain fart. Yeah, norovirus, rotavirus. So what do these things all have in common? They're caused by a pathogen, right? So communicable diseases are typically ones that are due to a pathogen of some sort, some kind of microorganism. And it can be direct or indirect transmission, which we'll talk about that again. And then what are some examples of things that are contagious?
Staph?
Kind of, in the right circumstances. Like I've heard of staph outbreaks happening on like wrestling teams because they're all using the same mats and workout equipment and stuff, but that's not really that contagious. So it's actually kind of hard to get HIV because what do you have to do to get it?
Sex.
Right? Like you have to like either have sex with someone or you have to like share needles. That's like super intimate, right? So you have to have a pretty close contact, and so it's actually not that contagious. Although, if someone has a high viral load, they are very infectious. So at that point, they could spread it to someone more easily. It's still not a guarantee that they would spread it. And I think, again, that's one of the reasons why getting proper HIV treatment is so important, because you want to get to undetectable, so that the viral load is so low that not only are you feeling good, but you can't pass it. It's virtually zero that you can pass it on to someone if you're like undetectable. So HIV is not exactly contagious. It would depend on the situation. What about coronavirus?
Yeah.
It's probably pretty contagious, right? But it's not as contagious as we thought it was. I think we still don't know for sure. How is coronavirus transmitted primarily?
Droplets?
Droplets, and now they think maybe even airborne. Right? And the difference is that droplets are heavier, they fall faster, and so you have to be closer to someone to take in the droplets versus airborne. If I sneeze, I can hit Bailey back there. So coronavirus might be both. Like it might actually be airborne, but it's not really that much more contagious than like the flu. It's kind of similar, but I think they're still trying to figure it out. So my point is that someone just sneezed. Bailey! It's like you can't even have allergies anymore in peace. Like we were in a meeting the other day and someone had like a cough side effect from a medication, and she was like coughing, but it sounded like kind of gross, and I kept side-eyeing her, and then finally I was like, I'm sorry. I'm like, it's not the Rona, is it? And I was like, you just now like there's no privacy. It's like, you have the Rona. Okay. So most contagious diseases are like airborne or droplet, like some of the more contagious ones. Okay, non-communicable means it's not spread person to person. So what kind of diseases would that be? What is a disease you get but you don't give to someone else necessarily?
Would that be like the thing you can get from like cats? Food
poisoning. Food poisoning you don't typically can give to someone else, but I guess you could maybe. But actually it's not that transmissible at all. Like usually food poisoning stays in you. The cat parasite is not necessarily transmissible, but it is communicable because it's spread through a pathogen that is in animals. I heard someone say malaria? Malaria is communicable because there's a pathogen involved, so you are communicating it indirectly via the vector. It'd just be stuff like cancer that isn't caused by a virus anyway. Diabetes, heart disease, depression, like things that don't necessarily have a pathogen involved. But you guys are right, there are some pathogen-based diseases that aren't transmissible person to person. So like food poisoning usually isn't. The only time I've kind of heard of people getting food poisoning from each other is if, like sometimes if it's someone caring for that person, then they can come into contact with the feces, like if it's a mama or daddy caring for the baby that's sick. Or obviously food handlers can pass it to each other. And then certain foodborne illnesses are really contagious, like Shigella. Shigella can like take over like a daycare. Like you know how sometimes daycares get gross and they have to close down for a few days and they got to bleach everything? It's usually because there was a food situation. Okay. All right, symptoms are subjective. So that's what the patient tells you they feel. And then signs are objective. They are measurable. So what are some examples of symptoms? What are things a patient could tell you?
Headache.
Pain, headache,
Nausea.
They could say they're depressed, right? They could say they're having trouble sleeping. They could say they're anxious. And then what are some things that you would measure?
Blood pressure.
Yeah, blood pressure, vital signs, blood pressure, pulse, the pulse oximetry, oxygen levels, weight, height, temperature. You can do blood tests, right? So those are objective, like they literally just are what they are. They're not up for debate necessarily because they're things that you can measure reliably. But some things are signs and symptoms, right? Like a patient can be like, yo, I got a fever. And you can take their temperature and be like, yes. Or no. Sometimes they think they do and they don't. That's why it's subjective, right? So some things fit into both categories. Okay, the other thing you can look at is morbidity and mortality. I think most people understand mortality a little bit better. That's the death that occurs. So like we actually, like for coronavirus, it is very age dependent what the mortality is. So it really is hard to give an overall mortality rate because I think it's pretty low overall. It's like 2%. But if you're like over the age of like 65, it's way more than 2%. And that's why it's important to stratify that data by age and maybe even by gender or by comorbidities. Like look at, okay, people have diabetes or hypertension or whatever to really see what's there. But it doesn't mean that it's not serious. Like I think that's what kind of bothers me about the whole situation is they're like, it's only people with pre-existing conditions. Screw grandma. I'm like, I mean, I really do feel like people were like that. It was like Lord of the Flies at the beginning. They're like, just let them die. And I'm like, oh my God. Like, no, that's not how public health works. Like, you don't just let people die because you don't think they're worthy of care. They're like a lost cause. You know what I mean? Because a lot of people have pre-existing conditions. And so that's just not, like, to me, that's just ethically incorrect. So mortality is the death, and so the more mortality you have, the more severe you could consider that disease. And then morbidity is the state of being unhealthy or diseased, so it's a little bit more of the subjective aspect. So like I always think about like, is this something that you're likely to get hospitalized with because it's that bad, or are you going to miss a ton of work because of it? Like if you think about corona, if you get like diagnosed with it or whatever, you have to stay away like for 10 to 14 days or whatever it is. If you get exposed, like a true exposure, even if you test negative, you have to stay away. So that is an economic and social loss, and I think that goes under morbidity. So I don't even know that we can measure yet the morbidity of COVID, like the impact that it's had economically, but they're starting to see the effects that it's had. Okay, you can also discuss how much of the host is affected. So what is the involvement of this disease? Best case scenario is that it would be a local infection, right? That it would just happen in that little area. But unfortunately with COVID, it seems like it can be systemic. They're seeing effects on the heart, a lot of destruction in the lungs. People were getting weird skin and blood vessel, like vascular problems. So it tends to be systemic disease, even though it primarily affects the lungs. Focal infections are ones that kind of start in one point and end up somewhere else. And of course, I'm having a brain fart. I can't think of a good example right now. I'm thinking of things that like cause pneumonia, but can also cause meningitis. So like strep pneumoniae, pneumonia could become meningitis if the patient was at risk for that, I guess. Septicemia is having a bloodstream infection. And really a lot of the symptoms of sepsis come from your own immune system. instead of having a localized inflammatory response, you're having a system-wide inflammatory response. And then that causes you to potentially go into shock and have cardiorespiratory failure. Bacteremia is a potential risk factor for getting septicemia, because if you have bacteria in the blood, that increases your risk of sepsis occurring. Toxemia is where they find toxins in the blood, and sometimes that is necessary to say that it's that pathogen. So I actually just read, I don't know how I came across it. It was like a really sad little case report of this little girl in India who got diphtheria. And diphtheria is like pretty much unheard of over here because it's part of the tetanus vaccine. So when you get like the Tdap or the DTaP. And so she was 14 and she had diphtheria and she ended up dying from it because she actually went into like VTAC and they weren't able to like resolve it though. So she ended up having like cardiac arrest. And when they sent the bacteria off to get it tested, it had the genes for a toxin. So I think that that toxin was actually like affecting her heart too, in addition to her upper respiratory system. And then viremia is the presence of viruses in the blood. And so if you hear that like they're trying to do a viral load, like say for an HIV patient, you're trying to figure out how many copies of the virus are like circulating, because that's a good indicator of how well treatment is working, because that should be hopefully low if the treatment is working well. All right, so when they say that you have an infection, it means that you're colonized with a pathogen. Disease means a change in health status. So does being infected mean you have a disease? Not necessarily, right? Because people can be subclinical, which means they're not showing signs and symptoms. And I think this is the reason why maybe some of you have noticed that back in the day, they were more likely to call them STDs, which means sexually transmitted disease. But now a lot of the vernacular has switched to STI, which is sexually transmitted infection, because what do you guys know about a lot of sexually transmitted infections? Do they manifest?
No, most
Most of them are asymptomatic, right? Especially like for chlamydia and gonorrhea, but especially chlamydia. Chlamydia is very asymptomatic in a lot of people. And so just because you're infected doesn't mean you have a disease. And that's why they kind of, it seems like a small point, like, it's semantics, but, it kind of matters. So disease is the change in health status, and it can be due to an infection. But infection doesn't mean you actually have disease, like you might not have any signs and symptoms yet. The etiology is what's causing it. Do we always know that? No. Do you guys know what the word is for when you don't know what's causing something? It's what was that?
Idiopathic.
Idiopathic, yeah. So idiopathic means you don't know what's causing it. In our class, we usually know because it's pathogens. We're interested in the microbes. In chapter 15, we'll talk more about pathogenicity and virulence. So pathogenicity is the process by which a pathogen causes disease. And then virulence is how severe. So like if someone asked you like, how does strep throat cause disease? What would you guess based off of what you know about strep throat?
Like, those colonies in your throat.
It does what?
It like makes colonies in your throat.
Yeah, so it like colonizes your throat, right? Like the mucous membranes there. What are the signs and symptoms of strep throat typically? Sore throat, fever, cough sometimes, postnasal nastiness, phlegm, redness, pus pockets, swollen tonsils, right? It's kind of inflammatory, isn't it? So if someone asks you, like, how do you think strep throat actually causes disease? Well, it irritates the throat through an inflammatory process. And that could be due to toxins that strep has or other things that your body is responding to. And that's kind of the double-edged sword of the immune system. Like your body's just trying to get rid of it, but that itself has side effects. Virulence is the severity. So if someone asked you, like, what do you think about the virulence of strep? Based on what you know, what would you say? Do you feel like it's very severe, medium severe?
It can be.
It can be, right? It's not usually if you get treatment. It kind of depends. I would say low severity. Like you're probably not going to die from strep throat, but if it's untreated, you can get invasive strep disease. Like it can go to your heart and cause like, what do they call it, scarlet fever, like rheumatic fever. That used to be a thing back in the day more when we didn't have antibiotics. Okay, and like if you're a baby, strep throat could be, I guess, more virulent. But if I asked you about like coronavirus, the one that's circulating right now, you'd be like, it depends, right? It depends on your age, but it tends to be virulent. So I would probably say coronavirus is like low to moderate, right? So my point is that there's very few things that are like highly virulent, luckily for us. And some of those we've eradicated, like smallpox. Okay, you can describe disease by how long it lasts. So the other word for that is duration. So how long are you going to be dealing with this situation? What's the onset? So rapid onset and quick resolution is acute. Long-term is chronic. Subacute is sort of in the middle. So it's not super short, but it's not long either. And then latent is where it reactivates. So you are sign and symptom free for a while, but then it can pop up years later potentially. And a lot of viruses have latency. They're able to do that. Okay, I think we've kind of said before that I guess you can't definitively say that anything causes anything. What you typically are trying to do is either support or unsupport the null hypothesis. So the null hypothesis says this doesn't do anything. And then the opposite is that it does do something. And so that's kind of what risk factors are. Risk factors are things that increase your likelihood of getting a disease. And it changes potentially the course of disease. And some risk factors are modifiable. So they're things that you can change. So like, what are some things on this list that you could change?
Your habits.
You could change your habits and lifestyle, your nutrition, your occupation, your environmental exposure, maybe. It depends on what they mean by that, right? Like if you live in a big city, it might be hard to do it. And then the things that are non-modifiable are things that you cannot change about yourself necessarily. So what would those things be? Yeah, your gender, your age, your immune status, your genetic background. Those are things that are just you. You can't necessarily change them. And that's why, to me, it's kind of mean, to say the least, to be like, who cares if you have a predisposing condition? Like, it's like, no, dude, like, those are things you can't change about yourself. Like, that's why racism is so ridiculous, right? Because you literally have no choice of how you're born and what you look like. But some things you do have a little bit of a choice in, but there's still going to be genetic things that sometimes override even your best efforts. So basically what this little pie chart is showing is what are the major risk factors for getting HIV. And notice that probably the biggest one is exposure to HIV. So in the 80s when HIV was discovered, people were like freaking out because they're like, how do we get it? Like, is it just around us? Is it in the air? And then they realized, like, you have to have intimate contact with someone to get HIV. Like, you're not able to just get it casually. Then they didn't have retrovirals back in the day. So that greatly increased your chance of getting it because it wasn't preventable as much. And then engaging in risky sexual behavior. So there's nothing wrong with having sex. But if you're doing it in a way that's putting yourself at risk, then there's consequences associated with that your risk of getting certain things goes up. So this is showing some other risk factor modeling for tuberculosis. So again, in order for you to be at risk for tuberculosis, you have to get exposed to tuberculosis. And luckily here in the US, it's rare to get TB. Now, they've also linked it to poor nutrition, because poor nutrition is going to mean that you just have maybe a weaker constitution, a weaker immune system. And then AIDS is also highly indicative of a risk factor for getting TB. In fact, in some parts of the world, if your TB test is positive, they also recommend you get an HIV test because it's very unusual for a healthy immune system to get active TB. The other risk factors, and it makes sense if you know how TB is spread. It's a respiratory disease. Poor ventilation, so living in a crowded, small place, crowding. And then absence of BCG, which is the vaccine that's used for tuberculosis. Well, we don't use it here in the U.S. because it works sort of well, but it interferes with the skin test. So we don't like to use it here. So a predisposing or risk factor is something that's modifiable or non-modifiable that increases your likelihood of having a disease and can also affect the progression of your disease, like what actually happens. Okay. Another measurement or classification of disease is how common it is, the occurrence and the frequency. And that's something that I like to think about so that I don't go crazy. Because, I don't want to be all fearful of Ebola because the likelihood of me getting Ebola is, pretty much zero because I haven't gone anywhere where Ebola is, right? So you want to worry about things that maybe I guess you should worry about, but it's all about, to me, risk assessment. What is my risk for getting this disease? So how you measure how common something is, there's two ways that are frequent, and that's incidence and prevalence. If you ever hear, let's say you got hurt at work and you have to fill out an incident report or whatever, that means it happened that first time, right? So incident means the number of new cases. And you're going to do that for things that hopefully only are going to happen one time. So incidence makes sense for something like HIV. Okay, the incidence of new HIV infections. And then they take that data and they stratified it based off of gender, because gender is a confounder. It can cause there to be an association or make it look higher than it really is when males and females just have different physiological risk factors, social risk factors. And then they further stratify that by race and ethnicity. And that's not to point fingers. It's to prioritize what limited health resources you have. So if you see that in one population you're making good gains, you're making progress, they're seeing a reduction, but another population isn't, then you might wonder why. And then those are the priorities that you set. That's how you set your health priorities. Prevalence is the number of new and pre-existing cases. So that works better for things that are common, that are always kind of happening, like heart disease. And then you can also stratify that by gender or age, because those are going to be confounding factors that can influence what the results really mean. So prevalence is the number of people who have a disease at a specific time. So if I asked you right now, who has seasonal allergies? Right now, I'm not asking you, when you found out, and people would say, I do. Versus incidence is, how many people just found out. They're a new allergy person, okay? Or, how many people just got the flu for the first time or whatever. So that's the difference, is that it's the number of people who develop a disease versus the number of people that have a disease. And that's incidence versus prevalence. All right, I'm going to stop there. And we'll pick up next time. Okay, we'll take a break and then we'll see you guys in lab at 11:30. Have a good weekend.